Yes. So good afternoon all who are present in the class, in YouTube and yes, in the Zoom. Okay, so I hope I'm audible to everybody. I'm just, please let me know in the chat box if if you're facing any issues like you cannot see the screen or you cannot listen to me properly. Okay, so, okay, let us start the class today. What we are going to discuss. We are going to continue the day before yesterday's class, that is about a biotechnology and its applications. Okay, so biotechnological applications, in the previous class we have studied about agricultural applications. Can anybody mention what are the agricultural applications we have studied in biotechnology? Yes, of course, why I'm mentioning we have studied, there are plenty of applications in biotechnology, or especially in the agricultural field, but we have studied very few in that, right? So just try to mention in the chat box. Yes, right. So I can able to watch the YouTube also. Please, the people who are answering in the YouTube, just let me know. Okay. So the applications what we have studied are BT cotton, right? And then nematode resistant tobacco plants, flavor saver tomatoes, whatnot, many others examples we have studied. Okay, so in today's class, we are going to study the one more interesting application of biotechnology in a medicinal field. Okay, so before going to that, as usual, let us recall something from the previous class. Let me see how much you can answer for this. How many people can give the correct answer? The question goes like this. Bacillus thuringiensis strains have been used for designing a novel biofertilizers, or for designing the novel biometallurgical techniques, or for designing the mineralization process, or for designing a novel bioinsecticidal plants. So yes, now you can answer for this. BT, Bacillus thuringiensis, will be using for what? Are we using it as a biofertilizer or biometallurgical techniques, or for the biomineralization process? And option D, we are using this for, or using this as a bioinsecticidal plants. Okay, I'm not getting the answers yet. Yes, very good, very good. So, yes, here are the results. That is biometallurgical techniques. Are you really sure about it, JNV Mahi students? No. BT cotton, already we have discussed in the last class, previous class, right? So BT cotton, where we are going to stop the attack of a worms or the insects to the cotton plant by using a Bacillus thuringiensis. So always this BT cotton, which is being used as a bioinsecticidal plant. Okay, bioinsecticidal or biopesticidal plant, anything you can say. It is not a biometallurgical technique. Understood? I hope you got the thing, or I hope you got the answer which I have given. Now, next question. To continue with that, BT toxin kills the larva of a certain insect. Yes, it kills, but how? Option A, they're saying by binding the activated toxin on the midgut epithelial cells, creating pores and leading to swelling and then lysis. This is a one way of killing the larva by BT toxin. And second way, what they're saying is by stopping the transcription of larval cell. Third way is by altering the central dogma taking place in the cells of the gut of a larva. And D, by stopping a protein synthesis. So now again, I'm going to launch a poll. Just let me know how BT toxin is going to kill the larva. Yes, poll is here, and the people from my YouTube, you can answer me in the YouTube, okay? I can say the answers. Yes, for the Zoom, I have launched a poll. Please give the answer from the Zoom, by the poll, sorry. I didn't get the answer set. If you finish this question, and we can move to the interesting topic of today's class. Okay. Give the answer. Yes. Very good. Oh, if you know this, you might be knowing the previous one also, right? So why you have given the wrong answer? Is it by any mistake? Yep, the answer is here. You have given the correct answer. Yes, in the previous class we have discussed, right, already. Bt toxin, how it is killing the larva? It is going to affect the midgut epithelial cells. Midgut means, gut means what? From the mouth to the anus, the entire area we are calling as a gut. In that, the midgut is the inner layer of midgut. Let's say this is a midgut, and this inner layer of a midgut is made up of epithelial cells. Okay, so these epithelial cells, in that, pores will be created, and later it will get swell and lysis ultimately. This is how this Bt toxin will be killing the larva. Okay. So now, as I said, we are going to study the applications of biotechnology in medicinal field or in the medicines. Okay. So let us start. The first one. Yes. Before going to the each and individual medicine, I just want to tell you there are totally 30 recombinant therapeutics have been approved. Okay, there are totally 13 are approved, but out of this, 12 are being marketed in our India. There are totally 30 recombinant therapeutic things have been approved in India. Out of that, only 12 are being marketed because remaining are still under the process. Okay. So in this class or in your NCRT as it is given, what we are going to study in the biotechnology, we are not going to study all 30 medicines prepared by the biotechnology. Don't worry. Only 3 we are going to study. One is genetically engineered insulin and gene therapy and the molecular diagnosis. Okay. Let us start with the first one, that is a genetically engineered insulin. So whenever we are talking about the insulin, the one thing which flashes in our mind is the disease for which we will take insulin, that is a diabetes. So now out of 3 homes, every 2 home will be having a patients who are having a diabetes, right? Earlier it was not so popular, or earlier I can say earlier the diabetes was not present everywhere, but now wherever the place we go, there we can see at least one diabetic patient, right? So what earlier they were doing, what they were doing is they used to extract the insulin. Okay, what is the connection? Before going to that, what is the connection between this diabetic patient and the insulin? Can I get answers in chat box if you can tell? So here what happens? Diabetic patients means what? The patients who cannot able to produce a proper insulin or the required amount of insulin, and the glucose is not going to convert as a glycogen. If insulin is producing, now the extra glucose is going to convert as a glycogen. Okay. Otherwise, what happens? The glucose will be remained as it is. Okay. If the glucose is remaining as it is, then the carbohydrate level or the sugar level in our body will get increased. So that at that time, what the people will do is body is not able to produce a proper insulin, right? So we are going to take a insulin externally from through injections, through tablets like that. And this insulin earlier in the olden days, they used to extract from the pancreas of a pig and a cattle. But later they got to know, as we are extracting this from the other organisms like a cattle, pig, we are going to extract, right? These are going to give us the allergic reactions. So then they started thinking, can we extract from the human and to give? Or can we do any other magic for this? Yes, they found out a magic. That is a biotechnology. To create a insulin by using a biotechnology, and that insulin is called as a genetically engineered insulin. Okay, how they prepared this genetically engineered insulin? If you know the entire process of biotechnology, then this is very, very, very easy. I'll tell you, see, this, yes. Insulin, normal structure of insulin I'm going to draw here. This is a chain A. This is a chain B. There are two chains, and the next chain is a chain C. Usually for a pro-insulin, we know pro means before the actual form of a insulin, that is a pro-insulin, which has a chain A, chain B, chain C. If we are doing it along, I mean, if we are elongating it, A, C, B chain will be there. Okay. But whenever it is activating or this pro-insulin is being matured as an insulin, now C chain will get removed, only A chain, B chain will be there, A chain and B chain. Between these disulfide bridges or the bonds will be formed. This is our actual insulin. Between A chain and B chain, the bond has to form. Okay, in our body, this is how insulin is going to be formed. First, pro-insulin will be produced, and this pro-insulin, C chain will be removed, protein chains. Chain means what? A protein chain, B protein chain, C protein chain. This is a pro-insulin. Later, this C protein chain will be removed, and between A and B chain, disulfide bonds will be formed. Okay, this is a normal regular insulin which is going to produce in the human body. But now we have to create an insulin by the genetically engineering or by genetic engineering. How we can do? Can we prepare all A, B, C chains? No. What do we do by preparing C chain extra? That is a pro-insulin. Anyhow, it will not work in our body. So what we can do? We can create a protein chain of A, protein chain of B, and we can make them to get a bonds, right? So now we are ready to use the insulin. But how to create this A chain, B chain? This is what the question. Chalo, let us see in the next slide. Sorry. Yep. So now what we are going to do is, do you all remember how biotechnological process works? Yes. First, we are going to take a gene, a small gene, which is going to synthesize a chain A. And this small gene is going to be introduced into the plasmid. The small gene will be introduced into the plasmid, and this plasmid later introduced into the host. Okay, now this host is going to produce a insulin in this, because we have our A chain producing gene in the plasmid, right? This plasmid has been introduced into the host cell, like a E. coli. Now in the E. coli, chain A has been produced. Same thing, this is the gene A, or gene which produces a chain A. Now, gene which produces a chain B, this will be introduced into the plasmid again, and this plasmid will be, just a minute, this plasmid will be again introduced into the E. coli. This is an E. coli. Okay, I'm underlining it because it is a scientific name. Now this E. coli is going to produce a B chain. Okay, so by this biotechnological process, we know how to synthesize a protein. So we have synthesized a protein A, we have synthesized a protein B, or a protein chain A and a protein chain B. Now, the thing we are going to do is just take a 1A chain, take a 1A chain from this, and take a 1B chain from this. Now we are going to allow it to create a bond, disulfide bond. Now this is a genetically engineered insulin, or genetically prepared insulin, right? So from the human itself, we have extracted a gene which is going to produce a chain A. And we have also extracted a gene which is going to produce a chain B. We have introduced them, both of the genes, in a separate plasmid, and these plasmids have been introduced into the bacteria. Now this bacteria is going to produce a chain A, chain B. Take those chain A, chain B and make them create a bond. Now we have got an insulin and go for a testing and then marketing. That's it. So this is how we got it. So first time in the 1983, right? Somebody has to do anything first time, right? So then only we can continue. This genetically engineered insulin also the American company has been prepared in the year of 1983. And that company is a Eli Lilly. Eli Lilly is a company. They have created a first human insulin in the year of 1983. Okay. And I told you how exactly it is going to happen. See, the disulfide bonds, they will make to create or they will make to form. Is it clear? Give the thumbs up if it is clear. Any doubts in the Zoom or any doubts in the YouTube? Any doubts? No doubts. Is anybody sleeping? Please don't do that. It is afternoon 3 o'clock. We had lunch and just now we came, ma'am. Okay. So one major important application. So by this, why we have to prepare by this? This is a quite costly, right? But just compare, if we are taking the insulin or if we are extracting the insulin from the cattle and the pigs, or pancreas of a cattle and pig, obviously that is the we are extracting from the other organism that is going to act as a foreign particle in our body. So some allergic reactions will be happen in the body. So to avoid that, we are creating a genetically engineered insulin that is called as a humulin. That is called as what? Humulin. So this genetically engineered insulin is gonna prepared by, first time it is prepared by the Eli Lilly company in the year of 1983. What they did? They have taken the gene which produces a chain A and the gene which produces a chain B. Then biotechnological process we have studied in the last class, right? So just by that, by the gene, we are going to get a chain A, chain B and combine both of them by creating disulfide bond. Done. We have got the insulin. Go for a testing, animal testing, human testing, whatever, all the testings. And then if any preservatives to be added, yes, for the medicines also we'll be adding a preservatives. That is why all the medicines usually they will be having some expiry date. Same thing, no, if we are taking any food, any processed food from the supermarket, obviously it will be having a one expiry date on it because we are adding a preservatives to this. The same way, all the medicines which are being produced in the any pharmaceutical company, we have to add a preservatives to this. Otherwise, they will be reacting to the bottle in which it is there, or they'll be reacting to the environment if we are opening it. So that is why we are going to add the preservatives, and that preservative will be having some particular date. After that, we cannot use that, or we should not use that, because preservative effect will be decreasing and the actual medicine starts to react with the environment or react with the things which are surrounding to it. That is why it is not recommended to have the medicines after the expiry date is being over. Okay. So done. This is about the genetically engineered insulin. Okay. So which of the following statement is not correct? This is a question. Let me read out statements for you. The proinsulin has extra peptide called a C peptide. Peptide means what? A protein chain. Okay. And then the functional insulin has a and a B chain linked together by hydrogen bonds. Genetically engineered insulin is produced by a E. coli. In a man, insulin is synthesized as a proinsulin. So which is not correct? Read the question carefully, especially in the biology. We need a more patience to read the question and the statements. The reason being is like it feels very easy, but whenever you are writing the question or whenever you're taking the question, there comes a problem. Okay, so which is not correct? Here is the option. Proinsulin has then extra peptide chain called a C-peptide. Is it correct or not? Like that, read all the 4 sentences and give me the answer in the poll. The people who are in the Zoom, just please answer me in the poll, and the people who are in YouTube, please answer in the chat box. I can see it. One more is there to answer it. To answer. Okay. I'm ending the poll. And which is the correct answer over here? See, somebody has given an option D. In a man, insulin is synthesized as a proinsulin. This is an absolutely true statement. As I said already, it is going to be synthesized as a proinsulin by producing a A chain, B chain. Between A chain and B chain, there is a C chain. This is the proinsulin. Later, this proinsulin will be converted as a chain and B chain with a disulfide bonds, right? So this is going to be called as a insulin now. So C peptide chain is present in the proinsulin, and in man, insulin is synthesized as a proinsulin. These two are correct. A and D statements are correct. Now let us talk about a C and B. C statement, genetically engineered insulin is produced in the E. coli. Do you all agree? Yes. A chain and B chain separately are being synthesized in the E. coli. C chain is also correct. Now, B, the functional insulin has A and a B chains linked together by hydrogen bond. Is it correct? No. A chain and a B chain both are linked together by the disulfide bonds. So this is the wrong statement which we have to choose, because they asked us which of the following statement is not correct. Is it clear to every one of you? Is it clear? Next is a gene therapy. Gene therapy means what? Some therapy we are going to do for the gene. Okay, like your answer, you will write, no, sometimes gene therapy for the gene. This is the definition. No. Yes, therapy we are doing, but what kind of therapy we are doing to this gene? That is what the question. Let us say there is any defect in the gene. We are going to change that defect. Either we are going to remove that gene and we are going to add the new gene. This is what a gene therapy. One example I'll take and I'll tell you, then you will understand very easily. You can see this girl, no? In the year of 1990, there was a 4-year-old girl, what you can see in the image right now. She cannot able to produce one enzyme, which is called a adenosine deaminase. Adenosine deaminase enzyme was not able to produce by this girl whenever she was very young. So if this enzyme is not being produced, the immune system cannot function properly. Okay, this enzyme, why this ADA enzyme is required? For the active function of an immune system. Now, why this enzyme is not producing? That is a question. Because there is a gene called as a ADA gene. There is a gene called a ADA gene. This gene is a defective here. For that, what they are doing is, right, we have to change the defective gene. What they will be taking? They are going to take the lymphocyte from this female or from the baby, 4-year-old girl. They'll be taking a lymphocyte and they are introducing this ADA gene to that lymphocyte and again keeping back those lymphocytes into the female body. Now what happens? she has this ADA gene. Now, by this ADA gene, she is going to produce the adenosine deaminase. And of course, her immune system will work properly. Here, what is happening? There is a one gene which is not working. So we are going to repair that gene by removing that and keeping the new one. Let's say if something is happening, like our TV is not working properly or our motorcycle is not working properly, what we'll do? We'll see first which is the part is not working. Okay, we understood this part has got damaged completely. We have to replace the part. Same thing. We are going to identify what is the problem. Okay, adenosine deaminase enzyme is not producing. But why it is not producing? Because of the defectiveness in ADA gene. And okay, we have to replace the part, which means the gene. So we'll be taking the lymphocytes out, keep the ADA, proper ADA gene into the lymphocyte and put this in lymphocyte back into the girl. Now what happens? The girl body is having a lymphocyte with the ADA gene. Now this ADA gene can produce a adenosine deaminase. That's it. This is called as a gene therapy. Is it clear? Is it clear to every one of you? Okay. So in short, they have given like this. But one thing you have to remember, what is this here? They did this gene therapy for the 4-year-old girl. Already the cells have been matured and everything formed. If they are doing the gene therapy for the younger people, or the ones the person has born at that time, if you are doing any therapy for them, you have to do it repetitively. Why to do it repetitively? The only reason being here is that they cannot produce the same gene again in the body. We have to produce it from outside all the time. But if we are doing it during the embryonic stage, all the cells are embryonic stages only. Sorry, embryonic cells is only during the embryonic stage. So we are just going to introduce the gene in that. At that time, no need to do it repetitively. That is a permanent treatment. But once the person has been born and we are doing the gene therapy, that is not a permanent one. Okay? Is it clear? Yes. So the last topic in this is a molecular diagnosis. Diagnosis means what? Just to identify the disease. To identify the disease also, we can use the biotechnology. But you can say, ma'am, why to use the biotechnology for the identification of disease? As we already have the urine test, blood test, serum test, all these tests we have. No. Then still why we have to go for the molecular diagnosis only? The only reason here is that all these urine test, serum test, blood test, all these tests we can understand or we can diagnose the disease once the disease is at the severe level. Okay. But if you want to do the diagnosis of the disease in an early stage, you cannot do by the urine test or a blood test. At that time, we have to check the DNA of our own body. Then only we will understand, okay, this person has this disease, either DNA or any biomolecules we have to analyze. Then only you can understand, okay, the person is having this disease. If you want to diagnose the disease in the early stages. Okay, you have some doubts, like I just want to go for it. I'm just having some doubt, like I may have this disease. So what you can do? Urine test, blood test will not give unless until the disease is in the next level. But if the disease is in the early level, at that time you have to go for the molecular diagnosis. What are those molecular diagnoses? First of all, how to go? Urine test, blood test, we all know. But molecular diagnosis means how to do. One is there, that is a PCR. Okay, so PCR, we all are in the era of a COVID now, right? COVID. We know RT-PCR, whether we are diagnosing the disease or not. Why we will go for RT-PCR? To treat the disease? No, to identify the disease, whether the disease is there or not. RT-PCR, real-time polymerase chain reaction or reverse transcription polymerase chain reaction. What is this PCR? Okay, RT-PCR for the like some viruses, retroviruses will be using a RT-PCR. Normally, let us talk about a PCR. PCR, what we will do here is, we are going to take a small piece of DNA and we should do the multiple copies of this DNA just to analyze it later. Because just with that one piece, we can't take any risk, right? With that one piece, we cannot take any risk at all. We have to make a multiple copies of it so that if one piece is being missed or one piece is being wasted, then we can use the another piece. Okay. So polymerase chain reaction, same thing is going to work. You can see the PCR tubes over here. There are a PCR tubes. In this PCR tubes, we are going to add the piece of DNA or the entire DNA which we want to multiply. The DNA, buffer solution, enzyme. Okay, enzyme which is a Tag polymerase enzyme. Remember this? Tag polymerase. We'll be using a Tag polymerase enzyme. So this enzyme and nucleotides, RNA primer, everything we are going to, you all know the DNA replication, right? So primer is required, nucleotides are required, and most of the enzymes are also required. But for all those enzymes, we are going to add only the Tag polymerase just to sustain higher temperatures. Because the normal enzymes which are used for the DNA replication, they cannot sustain for the higher temperature. That is why we will be adding a Tag polymerase enzyme which can sustain for the higher temperature. So add all these into the PCR tube. That's it. There you can see on the top of this machine here, keep this tubes and you give the how many cycles you want, or you give how many number of DNA copies you want. It will make a cycle. For one cycle, we are going to get from one piece of DNA 2, then 4, then 8, then 4 to 8, 8 to 16, 16 to 32. Like that, the copies are going to be multiplied and we can get a millions of DNA copies. Okay. And all the uses of PCR, where all we can use the PCR. One is to detect a HIV in the AIDS patient and to detect a mutation. What kind of mutation is there in the gene which is causing a cancer or whatever it is. If to detect a mutation, we are going to use this. And yes, there are many more other uses of this PCR also. Okay. Next is one more diagnosis. One is we can diagnose the disease by a PCR and one more is by a ELISA. That is an enzyme-linked immunosorbent assay. Okay, this is a very simple. Let's say any virus has been entered into the body, our body. At that time, body, yes, of course, we are being affected, but still body wants to fight against it. What our body will do? It is going to produce antibodies against that virus. Okay, so in our body already some antibodies are there, right? So what we will do if I want to know whether I have a disease or not, and my body is already having antibodies against a virus. So what I will do, I'm going to give some of my body serum or a blood to the laboratory condition. So what they will do is like, they will be having a one well. Okay, this is a question. They will be having a one well, which is going to have the virus here. Okay, which is going to have the virus over here. Now they will be pouring the sample which they have collected from me. If my sample is having an antibody, that antibody will come and bind to this virus just to fight against it, right? So if it is binding, which shows that my body is having the antibodies. If my body is having antibodies, it is very clear that already there is a virus in my body. This is how we can identify whether the person is having a disease or not. This is called as a enzyme-linked immunosorbent assay. Is it clear? Yes. So here is a question. The first clinical gene therapy was given in the 1990s to a 4-year-old girl with an enzyme deficiency of adenosine deaminase, tyrosine oxidase, monoamine oxidase, and glutamate dehydrogenase. Give me the answer in a chat box. Sorry, poll. The students who are in a Zoom, give me the answer in the poll, and students from a YouTube can give the answer in the chat box. Gene therapy has done first time on which enzyme? This is very easy. Answer quickly. Okay, I'm ending the poll. And the answer is adenosine deaminase. Okay. So that's it for today. Actually, what we have studied, let me just recall once again. Today, in today's class, we have studied about the biotechnological applications in the medicinal field. And these biotechnological applications are, one is biotechnological applications in that one is the production of insulin, genetically engineered insulin, and then the production of genetically engineered insulin, and then the production of what? Gene therapy. And after the gene therapy, molecular diagnosis. Okay, these are all the things we have studied in today's class, and the next class we are going to study some more things. Okay.